Today we're going to talk about the theory of gram staining. It is important to know that bacteria are very diverse. However, at a visual level, bacteria can look very similar. One means of differentiating bacteria is based on the peptidoglycan content in the cell wall. Bacteria which have large amounts of peptidoglycan are said to be gram-positive, whereas those with small amounts are said to be gram-negative.
As a result, when we perform a gram staining procedure, gram-positive cells retain the gram stain and gram-negative cells do not.
There are 5 basic steps to a gram stain: fixation, which includes the application of heat; staining with crystal violet; fixation of the stained bacteria using grams iodine; washing unbound stain with ethanol; and finally, the counterstain using aqueous fusion. To determine the validity of the staining procedure, a putative positive and negative control are smeared on the same slide as our sample of interest. In this example, the S stands for our sample of interest. The first step in gram staining is fixation, in which the back side of a glass slide containing the bacterial smears is passed through a heat source, such as a Bunsen burner. Bacterial samples are first exposed to the primary stain, crystal violet, which will stain both gram-positive and gram-negative bacteria. Next, grams iodine is added to the sample, where it will enter the cell and form a large complex with crystal violet. Ethanol is then added, removing much of the lipid bilayer present in the outermost layer of bacteria. The increased peptidoglycan content in gram-positive cells retains barrier integrity, despite ethanol treatment, whereas gram-negative bacteria have a decreased membrane integrity, which allows for the removal of the crystal violet iodine complex, thus removing the stain in gram-negative bacteria. To reiterate, after ethanol treatment, gram-positive cells remain purple, whereas gram-negative cells will not have stain. Next, we add aqueous fusion counterstain in order to visualize gram-negative bacteria. Without a stain, it is very challenging to see bacteria under the microscope. After adding aqueous fusion stain to the sample, the gram-positive bacteria will remain purple, whereas the gram-negative samples will be pink. When viewed under a microscope, ensure your positive control cells are purple, while your negative control appears pink. Upon confirmation of the validity of the stain, you can observe your sample to determine whether it is gram-positive or gram-negative. To perform a gram stain, you will need a Bunsen burner, a waste bin, a squirt bottle with water, an inoculating loop, a bacterial sample, a glass slide, and your gram staining reagents. First, add a small drop of water to the glass slide. Next, take a small amount of bacterial sample and mix it with the drop of water. Let the sample and water sit for 5 to 10 minutes until dry. Pass the glass slide with the bacterial sample through a Bunsen burner to fix the sample to the slide. Once fixed, add a drop of crystal violet to your sample and let it sit for 1 minute. After 1 minute, wash the samples with water. Next, add a drop of grams iodine to each sample and let it sit for 1 minute. Again, wash with water after 1 minute. Next, add a few drops of 95% ethanol to each sample and let it sit for 30 seconds. After 30 seconds, wash the ethanol off with water. Lastly, add aqueous fusion counterstain to each sample and wash off with water after 45 seconds. Your sample is now ready to be viewed under a microscope.